Hello, I'm Dr. Arthur Brodsky, Assistant Director of Scientific Content at the Cancer Research Institute. And today I'm grateful to be joined by Dr. Ivan Zanoni, a CRI Lloyd J STAR at Boston Children's Hospital and Harvard Medical School. Welcome, Dr. Zanoni.
Thank you for having me.
So your CRI STAR work focuses on a group of immune cells known as neutrophils. What are neutrophils, and what do we know about the roles that they play in immune responses, especially in the context of cancer?
So neutrophils are the most abundant cell type in our blood, and they are basically involved in every immune response. And during every inflammatory process, they play very important roles. And they respond very rapidly to an invasion of a pathogen or to a tissue insult. And they do this to protect us against pathogen invasion. And for this reason, they can produce many different molecules, mediators, that potentially contain a possible pathogen, but at the same time also may affect and alter our own tissues. Now, tumor development since several years is now seen as a type of inflammatory disease in which the immune system plays a very, very fundamental part. And the immune system works as a two-edged sword, and the same do neutrophils. So these cells can play anti-tumoral roles, but they can also become pro-tumoral. And the tumors really produce a lot of molecules that we don't fully understand yet, that are able to change this activity of the neutrophils from anti-tumoral to pro-tumoral. And so that's why their biology is so important to study. Ideally, if we can revert their function back to anti-tumoral, we may use a very potent ally against tumors.
That's interesting. I think that's a really important point you mentioned, that, we think of the immune system as purely protective, but because of how powerful the immune system is, it also has to have a way to kind of turn itself off, which, as you know, in cancer can sometimes go awry. So I think it's really, and as you mentioned, there's so many factors, and we don't quite know what tips the balance. So you're exploring a lot of these different factors that affect neutrophils activity. And recently you found that molecules known as type 3 interferons can impact neutrophils in a way that promotes the development and growth of tumors, specifically colorectal cancer tumors. Fortunately, though, you also found that blocking the activity of these type 3 interferons could disrupt this effect. So why is this information important, and how might you build on it moving forward?
Yes, exactly. So the growth of tumors is very complicated. There are many factors that are involved. The tumors themselves can produce different molecules or can alter their microenvironment. So not only the tumor cells, but also what is around these cells. And we are particularly focused on colorectal cancer, as you mentioned. And that's a very unique environment because it's in our guts. And in our gut, there are many microorganisms that live with us. They are called the microbiota, but they can also induce inflammation if they enter in contact with the immune cells. So it's a very unique environment. And the colorectal tumors learned how to grow in this context. And one of the molecules that is most specific of these tissues, like mucosal barriers, like the gut or the lungs, but let's talk about the gut, are these interferons, these type 3 interferons. And these type 3 interferons have a very potent antiviral role in the gut. Mainly look at that. So we were working on that. We were working on colitis. We were working on colorectal cancer, and we asked how these interferons that are called type 3 interferons, because there are 3 different families, may affect specifically the response of neutrophils during colorectal cancer. And as you said, we found that if we get rid of the capacity of these interferons to change the activity of neutrophils, we protect against colorectal cancer. Meaning that now we can potentially target, to come to your question, how we want to exploit it, we can potentially target this pathway in neutrophils, exactly to do what we were saying before, to revert neutrophils from pro-tumoral cells to anti-tumoral cells. And so we are studying this both in mouse models in vivo and in vitro, and we identified a specific type of neutrophils that play this pro-tumoral roles, and whose activity is changed when they do not see any more type 3 interference.
That's great to hear. I look forward to seeing your progress and how that approach goes. So in addition to type 3 interferons, you're also looking at how certain fat molecules, which are called oxidized lipids, how they impact the metabolism of neutrophils and subsequently the overall level of inflammation within our gut. What do you hope that this avenue of investigation might yield?
Yes, in recent years, we went back to the metabolism of the immune cells. So the immune response is very complicated. There are so many different aspects of it as it is for the tumor growth. And many groups, among those my group, really start to look how during an inflammatory response, the metabolism of different immune cells may change. And these changes in the metabolism really affect the capacity of the cells to respond and to exert specific functions. So again, going back to this unique environment in the gut, we know that during colorectal cancer development, very often there is an inflammatory process that is going on. And so we know that among the molecules that are present there are these lipids that are oxidized because of the inflammatory process. And studying other cell types before, we found that these lipids can change the activity of several types of immune cells. And now we are interested in understanding if these oxidized lipids may also affect the biology of neutrophils in the context of colorectal cancer. And we have preliminary studies that demonstrate that this is the case, and then how these phospholipids, these oxidized lipids may affect really the pro or anti-tumoral functions of neutrophils. We are actively looking into this right now.
Very interesting. And it's we've been talking about, I guess, two specific areas, the interferons and the lipids that you're looking at, but the gut, and all tissues are complicated, but especially the gut. You have microbes, you have our food, you have all these nutrients coming in, you have all our immune cells, you have the epithelial cells. It's a very complicated situation, as you alluded to. So as you're trying to kind of explore this situation and determine the mechanism and what are the causes and effects, what are some of the biggest challenges with respect to answering these questions? And are there any technological hurdles that you're addressing at the same time in order to get to these answers?
Yes, the system is very complicated, and we, for a long period of time, we thought to study immune cells like in a void, and so we gave only one stimulus, and then we look at how they responded. But now it's clear that the situation is way more complicated, and these cells receive multiple stimuli. And so this is what we want to do to understand how these cells, like the neutrophils, respond in this very complex environment in which they will receive multiple information from the tumor, from the inflammation, from the bugs that live in our gut, from these oxidized lipids. Of course, we need a lot of technology to do this type of studies, and there are many new technologies that really help us to do this. There is single cell RNA sequencing and many others. And we are intensely collaborating with other groups to further develop other technologies. We are working with people at Northeastern to develop a single cell proteomics, thinking really to move it towards the study of these immune cells in the context of the tumor. So there are technical challenges also because neutrophils are very unique cells. They die very fast in vitro. So it's hard to study them. It's hard to modify their genetic. I think that we can do with other cells. For other cell types. So there are many technological hurdles, but really the technology is advancing very fast, and we are trying to use everything is out there and help developing more to try to understand the biology of these cells.
Is it reasonable to assume also that, you know, as you develop these new technologies that allow you to investigate the cells in new ways, that maybe it could have other applications beyond just cancer biology?
Yeah, absolutely. That's the beauty of the technology, that sometimes you develop something for a completely different reason, and then it is repurposed for other reasons. But this is part of what is happening constantly everywhere in the world.
That's great to hear. So as a CRI STAR, you'll be receiving $1.25 million over the next 5 years to pursue your high risk, high reward research. Can you explain why this STAR funding is so important for you? And specifically, what will this support enable you to pursue that you wouldn't have been able to otherwise?
So it's a very unique type of funding. Our research is extremely expensive, and we get money from foundations, so from the NIH, to perform our studies. But usually we write a project with some aims, some objectives that we want to reach, and we have to stick to that objectives very tightly, because we have been given money to do that type of research. But research is a very complicated field, and sometimes you have an idea, you follow an idea, you do some experiments, and you find something that was unexpected, but very interesting at the same time. And so the CRI STAR really gives us the opportunity to use the money that we have been given to do this type of discoveries and studies. So we have a very high risk project. We hypothesize some possible results, but we will have to test, to do the actual experiment, look at the data, and then with this grant, we have the possibility to really follow what will be most important and most interesting in our data. And it's a very unique funding source that is given to the researcher more than the project. And of course, we will work in the context of colorectal cancer, but we have this flexibility that is very important. We have already some new preliminary results that were completely unexpected, and that we are starting to dissect right now, thanks to the support from the CRI.
I think that's a really important point to appreciate, that yes, we have the evidence and we have hypotheses, and you go in a direction that you think will work, but you never really know until you do it. And then, like you said, you get there and you might come up with some new discovery that leads in a new direction. It's great that you'll be able to kind of follow the data and the science as it leads. So speaking of, what do you hope to accomplish over the next 5 years as a CRI STAR? And kind of just zooming out a little bit, how do you hope that your work will impact the field overall?
Yeah, exactly. It is linked to what I said before. So there's the freedom to really look at the data and being able to follow new possible directions related to this project, maybe not foreseen before. It's what it is really important. And what I would really like to do with this grant is to study how these cells work as hubs that receive multiple information, multiple stimuli, and change their responses according to the stimuli. And understanding this molecular mechanism, what we really think we will be able to do is to highlight new possible therapeutic targets. And so this is the real challenge that we have, but also what we believe have all the tools and preliminary data to do, understand exactly at the molecular level what is happening, and define new possible targets that can be really targeted in the next 5 years or so. And to talk for the field, it's more complicated. But this idea of the integration of the multiple stimuli, I think that it may apply to many different cell types. And we have this preliminary data showing that really, if we give our type 3 interferons, but then we also give these oxidized lipids, things change. And so starting to dissect this type of questions, I think will be very important. We are looking at this in neutrophils and colorectal cancer, but this may apply to other tissue, other type of tumors, other immune cell types.
Absolutely. Yeah. I mean, immunotherapy's made a lot of great strides in some cancer types, but other cancer types, especially like colorectal cancer, there hasn't been as much progress for the patient. So hopefully approaches like yours, we'll need new approaches and new targets and new strategies that hopefully work like yours will uncover, and we can help more patients.
That's the hope, and that's why we are working so hard to get our data.
Can't wait to see what you find. So I just want to thank you again very much for joining me today, Dr. Zanoni, and I wish you the best of luck in your research.
Thank you. It was a real pleasure.